to the slides. The contents, you can see there are a lot of things to discuss. You can see there are 26 slides. It's going to be a very long presentation. And I don't know how am I to make you all understand what I've read. Anyway, first one is introduction goals, introduction to group theories. We talk about group theory, genetic codes, cell cycle, multinucleated cells, algebraic graph theory, something new that I've learned, graph morphism, network dynamics and groupoid, something new about groupoid, cellular dynamic models via graph morphism. Frankly telling, but number 8, I don't really understand. But I'll just explain what I've read. Number 9 is about results and conclusion and references. Okay, the first one, this introduction, I've made two pages of introduction, is none from the paper. Because first I need to know what is actually the real meaning of these nucleotides, genes, genetics. So I've Googled and find few references, and I've wrote introduction on that. And sorry about the spelling, this R is missing up here. It's a double helix structure. You can see the double helix here, and you can find this inside the nucleus of living organisms cell. And it contains information, all types of information, how organisms develop, survive, and recreate. And these nucleotides, called genetic codes, made up of phosphate, sugar, and 4 types of nitrogen base: adenine, thymine, guanine, and cytosine. So we are actually going to talk about A, T, G, and C, the whole paper. But then there's one change, that this T will convert to U when it goes to RNA. So what we're going to talk about is mostly about RNA. And what is a RNA? Is a ribonucleic acid. It's a single helix structure, translated copy from the unzipped DNA. This DNA, when it unzips, it will make a copy. So the copy will be from the A will be translated to a new base called U, uracil, and then T to A, C to G, G to C. And this is actually the structure of RNA after it has been translated from DNA. And then this RNA will be translated we'll go through the process of translation, where the RNA will be transformed into polypeptide change of amino acid. So this A will translate to U will be translated to A, C to G. It means like a negative, for those who have seen negative photograph. When we take photograph last some years, long time back, we have negatives. So the RNA is like a negative. And then to reproduce, to reprint, it will change the negative to negative again, so make it positive. So this will happen in ribosome, which is outside the nucleus. The ribosome will read the RNA's 3 bases at a time, which we call triplet. And this is what actually we call as codon at a time. And then these 3 base triplets will create a polypeptide chain, and we have 20 types of amino acids. And how these things happen, how these triplets arrange, the protein synthesis, and synthesize and the interaction network among proteins. All these are what the whole paper discuss about using the group theory from abstract algebra. The main goal of the paper is to identify an alternative set of mathematical ideas to relate to biology, specifically system biology. So here there are some symmetry and symmetry breaking events happen. Either it's bilaterals or radial symmetry. We can see in the following slides. So the first, the paper starts with introduction to group theory. Actually, they have gone through some very basic things about group theory. I only summarized the one that is important here, because I think most of us understand about group theory. So first, the paper starts with a square card in R3 space, rotate 180 degrees on X, Y, and Z axis. So this is R1 rotates here on this axis, R2 and R3, and then Cayley table produced, where is E, R1, R2, and R3. And if I draw a line here, which I don't know how to do it on latex. Sorry, I'm just learning how to use latex. So I struggle a lot to create this slide. It took me days. If I draw a line here, you can see that this actually commutes. So this is an abelian group. So this is the one, this is the table that we're going to use as a reference in other things later in genetic codes and so on. A bit more on group theory. The paper also discuss about finite group. The finite group G have important application in molecular system biology. The most significant, the one they use in this paper is symmetric group Sn and cyclic group Zn. So some explanation on Sn. Sn is a set in a collection of permutation of set of n elements and has order n factorial. Zn is a subgroup of Sn that contains cyclic permutations and also presented as one rotation by multiples of 2 pi over n and 2 the group of integers modulo n. The next is a group point. So what is actually a group point will be discussed later in the slide. So group point is more general than a group and made up of a pair of G, mu. So where G is a set of elements and mu is a binary operation, it's useful in explaining networks. In this chapter, we're talking about the networks, protein-protein interaction networks, PPI. Later we'll talk about that. So there are two things involved here. One is transcriptome, the other one is interactome. Transcriptome is a set of all RNA transcripts, including coding and non-coding in an individual of a population of cells. This coding and non-coding, when DNA has been copied to make RNA, before it goes into translation process, some of the RNAs will be arranged as a code, some of the RNAs will be removed from that single helix structure. So that's coding and non-coding. So not all of them will be used in synthesis of protein. So that's the coding and non-coding. Interactome, the whole set of molecular interactions in the particular cell. So how this protein that synthesize, how they interact among them. Something like that, that's what I understand. Okay. Next is about the genetic code. When the genetic code condense is when these nucleotides is the E, the C, the G, and the U, how they form codons, the triplets. That's what you call the condense from all possibilities. There exist many symmetry breaking events. A codon, a triplet, is represented as an element in the product of 3 identical sets. So we have 4 nucleotides here, U, C, and E and G. So that's S1. Let's say S2 is the same. Again, we take this. S3 also same. So we have to do the multiplication between S1, S2, S3. That means we are multiplying this. It's actually a power 3. So 4 power 3, we'll get 64 possible triplets. The reason is because we need our body needs 20 amino acids. To make these 20 amino acids, we need more than 20 possible triplets. So if we take S1 times S2, 4 times 4, we only get 16 possible triplets. For with 16 possible triplets, we cannot make 20 amino acids. So that's why we can't use doublets. We cannot use dinucleotides. It's not enough. So we have to take 3 triplets. That's how the nature works. Nature, we use 4 power 3 triplets, and we get 64 possible triplets. And then this 64 possible triplets, later I will explain that a sum of the triplets will give you the same amino acid. So we won't get 64 amino acids, but we'll get 20 amino acids, where some of the triplets will give the same amino acids. Yeah. So that will be shown later in the next slide. So by using group called clean group. The explanation for clean group, the one I found, is a 4-element abelian group, isomorphic to the symmetries of the non-square rectangle in 2 space. So using this clean group, they have divided this this dinucleotides. Dinucleotides means 2 nucleotides combined into 2 subsets, M1 and M2. So the M1 is AC, CC, CU, and you can see all this here, and M2 is CA. M1 and M2, they are different. The different set here, we have 8 here. We call this octets. There's another 8 here, another octet. So what is about the M1 and M2? M1 doublets are degenerate triplet. M1 is called degenerate triplet. What is actually a degenerate triplet? I have searched for it. It's not in the paper. It's in the paper, but I didn't understand. But then later I searched for it, where the third base will not influence on the type of amino acid. For example, if you look at this, the first one, AC, when you see the first two, because in triplets in codon, we need three to form an amino acid. We need 3 nucleotides, but here there's only 2 dinucleotides. So when you see AC in front, no matter what is going to be the next, whether it's ACA or ACU, ACG, definitely we only get one type of amino acid. That AC will determine the amino acid itself. So the third nucleotide that's going to join here in M1 will not influence the type of amino acid. That's what we call degenerate triplet. And for the M2 is different. The third base that match with M2 will decide the type of amino acid. So in M2, the CA will not determine the amino acid yet. CAA, the third one, the A will form a different amino acid. CAC will form a different amino acid. CAU might be different, and CAG might be different. So the first one is degenerate. The second one is the third nucleotide will decide the type of amino acid. So this is the detailed part of biology and also related with some mathematics here. And then the researcher, the paper also talk about how they break these operations, the operators into the identity, alpha, beta, and gamma, and how does it work. So alpha is the exchange between E and C, can be going either way, U and G, and beta between E and U, and then C and G, and for gamma between E and G and U and C. So this happens in the cell actually, in the codes. And the next one is what they did is they took this operation here and put it in a Cayley table, this table 2, clean group table for the genetic code exchange operator. So they found out that when alpha and alpha, that means cancel out, you can see that here, but the alpha and beta will give gamma, and gamma and beta will give, alpha and beta will give gamma, example here, and alpha and beta here will give gamma. So if I draw a line here, we can see it's symmetrical. So that shows it's an isomorphic with one another. And then the paper actually go on further by creating this K4 by K4 4-dimensional hypercube tesseract. We actually created this to put this dinucleotides. See the AC. Each corner are the dinucleotides you can see. So the blue one here is octets lie on the blue plane. M2 lies on the green. I have to go between slides forward and back because I have to show some diagrams. So M1, UC, AC, CC, GC, you can see here, this one. All this will be on the blue plane, 3 planes here. And then the M2, CA, AA, AU, AG will be on the green plane. 3 planes here. So There's a reason why actually the paper only shade this one. I shade extra one green here so that I can see easily. About the M1 octets. M1 octets are unchangeable. Stay in blue. As I said earlier, that M1 is a degenerate triplet. So no matter what is the third triplet going to be, a third decoder is going to be, it will never make any change in the amino acid. But then when it goes through these operators, octets are unchangeable. They will stay in blue under the operation EE definitely and VE. But interchangeable move to green under the operation alpha alpha and or gamma alpha. Just one simple example. Let's say I take alpha alpha. I'm going to see how the M1 goes to M2 plane. Alpha alpha. Take this example. I'll take this one AC. So if we look at the operator, A will become C, and C will become A. So AC and then CA. So this one, the A will become C. C can be here or here or here. And then C will become A. So AC will become CA. You can see that AC changed to CA. From AC, it jumped to the green plane. That is through alpha alpha operation. But then if you try to do beta E operation, this AC will stay in the blue plane, but it will move spaces. For example, the A will change to C, but C will still the same because we're doing beta alpha. Beta is A to U, sorry, beta is A to U. So if I do A to U, doesn't change. You can see that AC becomes UC and still in the blue plane. That's what I have explained through the diagram actually. That's what has been told here. It took some time for me to understand this. 64 elements in the genetic code were represented by 64-dimensional hypercube. The codons were symmetry operations. Multiple vertices of the hypercube code. This code here is a verb. The multiple vertices of the hypercube will code for the same amino acid. So some of, as I told, some of the codes they might be different, but these different triplets will give same amino acid. This shows a surjective mapping. The 64-dimensional hyperspace is considered as an information space. So if time evolution is included, so 64 plus one more time, we get 65-dimensional information space-time manifold. So the existing genetic code evolved into its differential manifold. The next one we're going to see is the cell cycle, the multinucleated cell. Multinucleated cell means we have more than one nucleus in a cell. The cell cycle is an example of a natural application of group theory due to its cyclic symmetric nature. So they have found that in paper, in the research, they say that there are 4 cycles for the cell, G1, S, G2, M, and then back to G1 again. So the cycle will keep going. It's a kind of, later I'll show you the cycle. Just a rotation. So these are the cycle phases. The same symmetry concept of square used to picturize the cell. The square that we see in the earlier slide, that one is used to picturize the same condition what happens here. And this is the rotation operations of the cell, R0, R90, R180, and R27. G1 to G1, S to S, GS, G2 to G2. This is actually the identity. And this G1 to S, S to G2. This is the rotational on 90 degrees, 180 degrees, and 170 degrees. Then we draw the table here. And this table looks similar to the one that we have seen in the beginning of the slide. Where if I draw a line here, I can still see the group, the class, the cycle is abelian and isomorphic to the cyclic Z form. The paper also did some experiment. What they did was they transferred the nucleus from one cell to another cell, so to produce a multiple nuclei cell. So that's what the multinucleated cell means. You take one cell from, you take the nucleus from one cell and they insert into another cell which already have one nucleus. So you have two nuclei now. And then they want to see what is the effect. The question in mind, what effect would the G2 nucleus have when transplanted into a cell whose nucleus was in S space. So they actually transplant the nucleus here in this phase here, S2. So after they transplant into S2, they want to see what happened in the next phase, G2. That is what explained here. So the experiment were to identify chromosome condensation because they have two nucleus, so there are chromosomes on one nucleus and chromosomes on another nucleus. So they are going to combine, they're going to condense. So I think that's what they're trying to explain. Chromosome condensation and the regulation of the DNA synthesis. And then next, what they did, now they try to draw a Cayley table again. But when you look at this Cayley table, it's actually different from the earlier one. So what the researchers said is that we can't find group structure for the set G1, G2, Sm. So normally in a group, any row or column of the multiplication table will contain the element of G only once. For example, here, this table here, only once, each of them. That happen here. But then it fails in the S and the M. Look at the S column here, we can see, and the S row here and the M row here, so the S and M, it fails. And the product of G1, G2 is undefined. You can't define what is this product of G1, G2. It's G1, G2, S and M carries the structure of a group point. You see, it is still a group point. Later I'll explain what is the definition for group point. The next part is graph theory, graph morphism. So this graph theory is something that is a bit new that I've learned. It's a network graph theory. Network graph theory is a primary tool for system biology to explore molecular system dynamics on a network level through morphism concept and algebraic graph theory. The missing connection in protein-protein interactions is the PPI that I said just now. Protein-protein interaction. Protein and protein will interact among them and they will create a network. This PPI network can be found or the missing connection in the PPI network can be found or build vector matrix-based models representing dynamics of changing PPI network. This figure 3 here, I think I have to do something. I want to explain something of this. Look at the notes. Okay, I want to show something here. If you look at these black dots here are the protein nodes, and you can see the lines, the red lines here are the networks. So this is actually, it might look like a rambutan, but it's not a rambutan. It's actually nodes of proteins. They interact with each other, the networks. You can see the lines. So these networks are the one that they use, they picturize as this network graph theory. Actually, the protein-protein interaction network of this is the experiment done in the lab by previous researchers. So this paper actually just talk about this. This is done by someone else. So then they explain a bit about what is actually a network graph. Network graph G, B, E, where n vertices or nodes, n vertices, V, and set of edges links. So the V is vertices, E is the edges. So the vertices and the lines will be the edges. The graphs can be written as n by n adjacency matrix. I didn't show any matrix here, even in the graph, we didn't explain much about. Sorry, even in the paper, they didn't show that matrix, adjacency matrix. You can actually try to find this. Adjacency matrix A, where the non-diagonal ij entry, aij is the number of edges, vertex I to j, and the diagonal entry aii depends on the convention from vertex I to itself. Eigenvalues lambda I can be computed to produce the spectrum representing the network graph. Actually, there are a lot of mathematical models, mathematical formulas they use here to measure this, but they didn't show in calculation. They just say that these eigenvalues can be computed to produce the spectrum. And let's say that we can also find trace of the matrix, and the trace A equals to 0, trace A square equals to 2n, and the trace A cubed equals to 60. N is the number of edges, and T is the number of triangles in the graph. So incident matrix B, matrix having one row for each. Incident matrix is another matrix. There are two types of matrix. One is the adjacency matrix, another one is the matrix having one row of each vertex for each vertex and one column for each edge. So I'll show that in the following diagram. This diagram here is the line graph. This is the incidence matrix. Involves line graph LG, where the edges of the graph, edges of the group are the nodes. These edges here, this one edge here is considered as a node. You can see a dot here. So they're taking these edges as a node. This edge here is a node. This edge here is a node. So each edge will be a node. But then they will join this edge with this edge. Yeah. Join this if and only if the corresponding edges of G share an endpoint. Means this edge and this edge have the same endpoint here. So I can join this with the red line that will create the line graph. But if you look at this edge here and this edge here, these two edges, they don't connect at the same endpoint. They don't have a same endpoint. So there's no connection between this node to this node. The red represents the line graph, and that's what actually they're trying to explain in this. That each one row for each vertex and one column for one for the row and one for the column. And B is not a square matrix. B is the incident matrix. Incidence matrix. So B is not a square matrix like A. A is a square matrix, but B is not a square matrix. So the relationship between the adjacency matrix of LG and the incidence matrix of G is B transpose B equals to 2 identity minus A LG. So this is what is derived in the paper. So I just follow what they explain. I tried to test this. I tried to put some numbers and calculate, but I didn't really successfully figure this out. This allows to calculate any potential new matrix on some complex molecular networks. This is something that we found. The next one is about network dynamic and groupoid formalism. This is about groupoid. I said I'm going to explain about groupoid. So what is actually a groupoid? Groupoid is important to understand the dynamics of molecular interactome networks. interactome is the interaction between these protein nodes. What is actually a groupoid? Groupoids are algebraic objects that looks like groups, but the conventional group operation is undefined. That's what I found when I search and also in the paper. Nothing much more than this I can actually explain here. Groupoid approach the dynamic of small networks when they are directed graphs. Directed graphs, this graph, non-directed graph, but if there will be an arrow, a direction, there's an arrow here, let's say from here to here, there's a direction, that's called directed graph. So using groupoid approach the dynamic of a small network when they use a directed graph, they examine, and these small networks are wired together to make large networks called circuits. Biological neuron, the networks, nets made up of 2 and 3-dimensional array of frequency control oscillators, voltage control oscillators, and logic gates. These components has the ability to store memory in the form of dynamic patterns and image cycles. The next diagram here, you can see this. They explain more about what we talked here. So, the cyclic group Z4, Z4 is a simple description of a cell cycle. They use Z4 to explain cell cycle. This is a cycle that I said that I'll show. This, yeah, cycle. S to G2, G2 to M, and then back to G1 because it works. It goes in a circle. This. And Z4 upgraded to comprise the observation that G1 and G2 are metastable in the same cell. So they actually changed this. They upgrade the Z4 to another level. They show in this figure. I wrote wrongly. It's supposed to be figure. Yeah, figure 5, correct. This one. This multinucleated state could communicate with cancer cells. Says that this multinucleated state, the multinucleated, the one that we learned earlier just now, when they insert one nucleus into another cell, said this could communicate with cancer cells. So it's a kind of solution we found for cancer. And I know this is about this very shallow, not very detailed. Which the two nucleus, a pattern of similar central pattern generator (CPG) of C6. So they actually try to relate this with another CPG oscillator. Here, diagram of 6-node CPG. It's also called C6 network. They try to connect this. They try to find a connection, and they show that this works. It goes in a circle again, just like this. And we also explain that we consider about the C6 network with only one bit active. So we have 1, 2, 3, 4, 5, 6 here. 6 here. So we start with 0, 0, 0, 0, 0, 1. And it goes anti-clockwise, as I say here, anti-clockwise here. Anti-clockwise direction. Then the transition is 0, 0, 0, 0, 0, 1 will become 0, 0, 0, 0, 1, 0. And then that will become 1, 0, 0 here, then 1, 0, 0, 0. And then the 1 will move. You can see the 1 is moving to left. And after that, it will come back to the original position. So after 6 rotation, the ring dynamics is the same. So the C6 group table drawn here is actually similar to the multiplication table that is shown, and it shows the group is a billion. So when I draw a line here, you can see the group is a billion. Try to connect it. This is the last. Yes. This is the last part. Seriously, I don't really much understand about most of the things, but this is the last one that is most confusing. Dynamics models via graph morphisms. So they said in the research they found three problems with the PPI network. The figure 3, this PPI network, this rambutan shape here. The three problems are, the first is the biophysical interactions are believed not necessarily occurring in the actual organism. And they say the second one is the PPI network for the most organism constitute only about 10% of the actual PPI. And the third one, it is a static network where time do not vary. So they said if it's a static, then it's meaningless for the concept of life form. So what the research did is they tried to resolve this, the PPI in time by conducting a relational join between transcription and PPI data. I think only the biologists understand about this. Then they explained that on the previous experiment done, this previous experiment done by another researcher before them on the yeast, the one that we see on the figure 3 just now, they say that the previous experiment done on collecting transcription microarray data at every 5-minute interval. So they try to do at every 5-minute interval, and they say the experiment shows heat. There is a heat map that I didn't show here. Actually, they found out that is heat. After every 5 minutes, they can see the change in heat. So the heat is growing and then reducing and growing and reducing. So they say that something is going on actually. So it's not really static. The experiment shows heat map for most of the genes during the cell cycle. The map shows periodic structure. Sorry, there's suddenly two structures here. The map shows periodic structure and also explains periodicity in the transcriptome. Another experiment on the yeast, done on the yeast by another researcher. They say that they have some attention on resolving the proteome into molecular machine during the cell cycles. What is actually proteome? I've written down here. Proteome is the entire set of proteins expressed by a genome, cell, tissue, or organism at a certain time. The next question is, what is genome? Genome includes both the genes and the non-coding genes. Because when I do this research, I start asking questions, what is proteome? What is genome? So I try to state it here, what is actually proteome and what is genome. So the condition of the cell at a certain time is represented by the function, use the function at time. And the transcriptome and the PPI network will combine the view of the proteins and their connectivity as a function of time. So as I said earlier, every 5 minutes interval. The interactome details every 5 minutes interval. The diagram is in figure 6 here. It looks like this. We see here, you can see this, there are more blue color compared to red color. You can see the red colors started growing. You can see more red colors here. So this after 5 minutes, the gap is between 5 minutes, 10, 15, 20, 25, and then goes to 30. You can see start reducing. The rate reduce, and then start increasing again, and then reduce, and then increase, reduce, and increase. This is the cycle that they have said. This is the G1 cycle, this is the S cycle, the G2 cycle, the transition between G1 and M, and then go back to G1. So that's what they say here. The red notes shows disappearance rate, and the blue notes shows appearance rate. So this is more protein here. The proteins start reducing, and then start increasing again, and then start reduce again, and then start increase again. This happens in the cell cycle. But the real understanding of this, I think, I have to know more biology to understand the real meaning behind this. And next is the results and conclusion. So what I can find here is a few mathematical ideas in understanding biology of living systems were discussed. They focus on group theory, explaining the relationship between mathematics and the molecular systems biology. And in the genetic codes, there is a possibility of using perturbation theory, exploring the adjacent possibilities. 65-dimensional spacetime, 64 is for the types, the triplets, and 1 is time. So total is 65. The algebraic graph theory, the graph theory used to evaluate the PPI. The PPI is a protein-protein interaction. The diagram I showed the networks. In the network dynamics and groupoid formalism, the network graph used to understand the phase space of the network dynamic by simple C6 network. So they use the C6 network here. I don't know how they found this relationship, but this is the relationship they use to explain the network. There was also some conjecture at the ending of the paper that there might be a better way to carry out this might be this research. They found out that especially the multinucleotide, when they insert the nucleus into another cell, you have two nuclei. They said this is a way of treating discovery of a drug for treating cancer. And the last part is the references. There are 2 pages of references here. These are the references that I have. Apart from the paper I've reviewed, I've also searched a lot of internet, some journals, some books to understand a few things. Because even though it's some of them are maths, but some of them are something new, and most of them are biology. I think I'll stop sharing now. Done with the presentation. That's a long one.
Okay.
Any questions can be asked, but I don't know whether I can answer, but I'll try my best. Because it's something very, very, very new. The concept of the genetic code, so I took this paper. And then it gets harder at the ending. And it will take 21 pages.
There's a question by Ahmad.
Yes, Ahmad. Ahmad, please have mercy on me.
Silly question perhaps. So does this research is using the pure mathematics to understand some kind of biological?
Yes. Actually, this paper, I think, done by some mathematicians or physicists to understand this biology. Because if I look at the paper, they said it was started, this concept of the molecular system biology were actually tested. First, the DNA was founded by, have you heard of this Francis Crick, right? Francis Crick is, and James Watson. Francis Crick is a biologist. But then James Watson, Francis Crick is a physicist, and James Watson is a biologist. These 2 people, actually a physicist, work with biologists from DNA, the double helix structure. So this paper, mostly they talk on, as you said, yes, they use a mathematical concept to understand the cell cycles and the molecular system biology.
Okay, any other question? I have plenty of questions, but no, I'll let others question first.
Selvam, hi.
Yes, why?
I have one Gelson question.
Because regarding on the mathematical, because usually we can only ask on the mathematical side, I will not ask from the biological side. Yeah, because
actually, you know, the reason I take this paper because I know in biology I won't have many questions.
But the mathematical side was very clearly understood and explained by you. That was a really strong point. I wanted to ask one girlfriend question is regarding, because in the paper you mentioned regarding on the RNA DNA transcription with mathematical model as well as the cell growth cycle. That means the cell duplication cycle.
Yes.
Do you include, I mean, is there any research regarding on the specialization of somatic cell using mathematical model?
No, I didn't see any somatic cells in the paper.
Research yet?
Yeah, in the research, I don't see any somatic cells. I don't know whether if I missed it, but I've never seen a somatic cell.
Because I mean, I'm just wondering. Usually, for somatic cell, they eventually going to specialize into certain cell in our body, and the specialization actually, I'm not sure is there any mathematical model to be used to understand what probability of certain cells could be specialized by certain somatic cell. Since also is the same.
No. There's no somatic cell. I don't see any. I don't know whether if I missed it, but I'm not sure I've never seen.
Okay. I'm just curious on what.
Mainly on the nucleotides, on how the DNA to RNA, from RNA to amino acids.
All right. Thanks.
KY. KY. I thought you were asking mathematical question.
Because there might be a mathematical model to explain that process as well, since Selvam reviewing this paper is regarding the connection.
I think Ganesh has.
Yeah, Ganesh. Go on, Ganesh. You have to unmute yourself.
Okay. In one of your slides,
Yes.
I think in slide 6, you have trace of A is equal to 0, but later you have a trace of A square equal to 2. And how is it possible?
Okay. How I got it, I don't know, because the paper, that's all the paper explained. Yeah. That's the formula. That's the formula. But Ganesh, I have the same question when I read the paper and I tried doing it. I create a matrix. I create a spy matrix. And then I tried testing it, it works.
How?
Numerical values. I put some numerical values because for me to review the paper, I think I took long time. Days. So yeah, correct. The question you asked, yes, I have this question, but they didn't mention anything on that. But when I tested with some numerical values on matrices, and then I found out it works. It works. I get the same.
Okay.
I get the same. Yeah, please, you know, right? How to find the place, so the diagonal.
Can I
Chan.
add a little bit? The trace of A is basically the adjacency matrix. So basically it's always 0. So that is true. But when you trace A squared, right, that squared, actually the A squared means that you use the adjacency matrix and then you multiply it again with the adjacency matrix. So that's why you get a different value. You get the age.
Yes, exactly.
But actually, usually, if that means if you take any matrix, trace of a matrix, let's say trace of a matrix is 0, and then you can have a trace of A square can be different.
That represent I think the number of age, right? It's mentioned there. Yeah, number of ages. Yes.
Yeah, the diagonal. It's just the diagonal add up.
But it's not true for general matrix, right?
Are you asking whether the diagonals are 0?
No. I'm comparing it with the general matrix.
Oh.
So I don't know.
No, okay. I mean, even if trace A is 0, but no A squared, basically what you have is Aij Ji.
Yeah.
So you won't get unless all the entries of the matrix are 0, then otherwise.
the main diagonals entries are non-zeros.
Okay.
Non-zero. So they add up to 0. It means there might be some negative numbers there. There are some negative numbers.
Actually, the adjacency matrix represents the network. So if let's say the diagonal part is not 0, that means you will have a self-link, self-loop like that.
Right.
Yeah, basically in those kind of network, you do not have self-loop. So that's why the diagonal part is always 0.
Okay.
So if it's not 0, then you have a self-loop. It connects with itself.
It connects with itself, yes.
Okay.
Okay, any other question before I fire my own question?
Mark the paper, definitely.
Well, I've yet to do it. Okay. By the way, when you mentioned about the 60, Ahmad, you want to ask question first? Tekan. Okay, you mentioned about the 64-dimensional manifold. 64 or 64.
Triplets, sorry. Triplets.
But my guess is that really true, because essentially what you had was just like discrete points rather than a variable. No, just like if you take the three-dimensional space, then you think of x, y, z being continuously varying. But in your case, it's going to be a set of alphabets which will not vary, right? So it's a bit confusing in that sense that probably what you have is something like a discrete set of points rather than no continuous kind of space.
Can I rephrase? Are you talking about that the DNA, the RNA, the structure? Whether the triplets are taken after every 3 triplets, after every 3 nucleotides, immediately they go to the next 3. That means, are you asking whether they overlap? Something like that?
Okay, essentially what you have is this 4 to the power 3, right? Yeah, 4 to the power 3, but that 4 is a fixed 4, see? Okay, meaning it's not a variable. So if I want to say it's a 64 dimensional, then your 4 variable symbols, you probably have to make it as a variable. Okay, but is that the case here, or is not the case?
It's variable because, as Prof, you said, that these 4 are fixed.
Yeah,
The
so
UA, G, and C
that's
are
what I
fixed.
thought.
This
Yeah.
is what we have in the nucleus. They try to find the permutation between 4 of these nucleotides with another 4 and then another 4. They find the permutations to be 64. That's what I understood from this. So I don't think I really answered your question.
Okay. It's okay.
About the variables part.
Okay, it's all right. The other question was the transition. You know, when you had from the point of doing the 64 or 65 dimensional thing to the network. Is it totally independent, or is related in some way or another?
Because they found out that if this 64 permutations, they said that it won't happen only at a time, it will keep changing.
Okay.
When the cells start going through the cycle. So they added the one dimension of time into it. So that's what I have understood from the paper, and they said it's a 65 dimensional, and they say it's a manifold. I don't know much about the manifold part.
Okay. But then the networks are going to be more than 64 dimensional, right? 64 no, points. Okay. I'm running out of what you call memory to record anymore, so I'll stop recording.